Hi, good morning, everyone, and welcome to our Sickle Cell Data Collection Program for California's webinar series. My name is Niani Coker from Tracking California's SCDC program, and I will be today's moderator. We have a great webinar planned for you. But first, a few housekeeping items. I have muted all conference lines to eliminate background noise, and this session is being recorded. The recording link and slide deck will be made available and automatically sent to everyone that is registered for the webinar. We will be taking written questions through the chat box or question function on the control panel of this webinar, and we will hold our question and answer period after the presentation has ended. There are currently no updates at this time from our CDC partners or our California SCDC program, so we'll get straight into our main presentation on community-engaged education materials for sickle cell disease gene therapy. I'll be introducing today's speakers, which are Vence Bonham Jr., JD. He is an associate investigator in the National Human Genome Research Institute's Division of Intramural Research, Social and Behavioral Research Branch. He leads Health Disparities Unit, which investigates the equitable integration of new genomic knowledge and precision medicine into clinic settings. Research focuses primarily on social implications of new genomic knowledge, including the role of genomics in exacerbating or ameliorating racial and ethnic health inequities. His research group studies physical and mental health of individuals living with sickle cell disease. His group is currently focused on the study of equitable access to emerging gene therapy technologies. Mr. Bonham also serves as a senior advisor to the NHGRI Director on Genomics and Health Disparities, which complements his research program. He will also be co-presenting with Tiana Amini, who's a post-baccalaureate research fellow in the National Human Genome Research Institute's Division of Intramural Research, Social and Behavioral Research Branch. She is a part of the Health Disparities Unit as well, a group led by Mr. Bonham. Her research primarily focuses on the advancement of gene therapy and on the need for education and community-oriented methods to engage patient populations as gene therapy continues to expand. Tiana received her bachelor's at the University of Chicago, where she studied sociology and biology. Ms. Amini plans to go to medical school once she completes her fellowship. So I will turn it over to our speakers and allow them to share their slides now.
All right, thank you. So, well, I can't see. All right, okay. Here we go. All right, so can you see my screen now?
Not quite. Yes. Yes, I can.
Okay, well, good. First, I just want to say thank you again for this invitation to speak here as part of this webinar series. On behalf of my colleague, Ms. Amini, and myself, we are really excited to share with you some of the work that we're doing and to raise a number of questions for the community to consider. So the talk that we're giving today is Community Engaged Educational Materials for Sickle Cell Disease Gene Therapy. So let me frame this talk and what I would like to do in the next 35 to 45 minutes. First, I want to give some context of where we are today in 2021 with regards to the legacy of sickle cell disease and the context of thinking about these 111 years of legacy for this disease. We'll explore sickle cell disease gene therapy clinical trials and what's the current status of those trials. Where are we with regards to the number of trials within the United States and across the world, and the context of the different demographics of the individuals that are being recruited for those trials. And then I want to share with you some data from a study that my research group conducted, where we engaged adults, parents, and healthcare providers of individuals living with sickle cell disease to get their opinions with regards to gene therapies. And so I want to share their voices as part of the context of this discussion about educational materials. And finally, we want to really explore what is currently the status of gene therapy educational materials for sickle cell disease and why we think it's important to create community engaged gene therapy resources. So let's start with this history of this disease. And I want to just make a comment that this is just the Western medicine, US centric history of sickle cell disease, with a recognition that individuals have been managing this disease for thousands of years across the world and have described it in different ways. But I think when we talk about gene therapy today, we need to think about the last 111 years and what has happened with regards to treatments for sickle cell disease and the management of sickle cell disease over the last 111 years. And I often start a talk by talking about Walter Clement Noel. And Walter Clement Noel was a young man who was from Grenada, island in the Caribbean, who came to Chicago, Illinois to go to dental school. And I want to highlight he was coming to go to dental school. And it's the one context of an understanding of his disease. But when he arrived in Chicago, he started to have healthcare problems. And he was seen by a physician by the name of James Herrick. And James Herrick identified within his blood cells that some of his cells were oddly shaped or sickle shaped. And so James Herrick first identified sickle cell disease within the Western medicine context. This was published in 1910 in an article as a case study in the Archives of Internal Medicine. Mr. Noel was followed for a number of years while he was a dental student in Chicago. But Mr. Noel returned to his island of Grenada and was a dentist there for a number of years before he died. And you'll see here an image from the cemetery where he is buried with the recognition of Mr. Noel. However, there just lacks any pictures of Mr. Noel, but I think it's important to think about where we are today and curative therapies and the history and Mr. Noel and Dr. Herrick. So the frame of our talk today is focused on innovation and thinking about where we are with regards to sickle cell disease treatment today in 2021 and the issue of innovation. And I argue this is a very exciting time for the treatment of sickle cell disease and particularly the potential of curative therapies. And so the framing of thinking about where the science has moved with regards to potential new treatments for sickle cell disease and the innovation with regards to these treatments, we argue we need to do the same with regards to resources to help individuals living with the disease and their families and their providers understand these new treatments and technologies and their importance with regards to the care of individuals with sickle cell disease. There's been an explosion of scientific literature around gene editing, gene therapy related to sickle cell disease. This is an exciting time, and I argue we are entering a new horizon of great hope with regards to curative treatments. And so a recognition of that requires us to think about what do we do. So this article from John Tisdale and Suile Thien has a really nice figure that really talks about current and future treatments for sickle cell anemia, thinking about drug treatment, allogeneic transplant, ex vivo gene therapy, and ultimately gene editing and in vivo gene therapy. And so thinking about all of these different treatments that are now potentially available over the next horizon of 10 to 15 years for individuals in actually managing their disease. But today, with regards to the future treatments of the gene therapy, both ex vivo and ultimately in vivo, we are still in clinical trials and at an early stage. And so what I would like to do is to go through this slide that I received from Dr. John Tisdale, and I want to just acknowledge him for this slide that really does a nice job of really framing the clinical trials that are currently in place with regards to gene therapy and sickle cell disease. And so let's just start here. You'll see that the first gene addition trial, that is addition of a gene versus editing, started in July of 2013. And you see they're continuing now. So in 2013, with editing trials opened, and then in 2018, gene editing trials opened. With regards to the phase 1 safety studies, you see here on those trials that are in phase 1 with regards to determining both the efficacy as well as the safety of these new treatments, and then the number of participants enrolled. So you see at the time of the preparation of this slide, the number of individuals who are actually participating in these clinical trials is fairly small. And a recognition that for us to move forward this field, that there are going to have to be more individuals living with sickle cell disease that decide to participate in these trials. And so this issue of recruitment to participate in trials is an important component of the education phase of clinical trials and clinical research. And so the numbers in the trial, the numbers completed, you see here the number of individuals who have completed the trials is very, very small. And you look at the age of the individuals, you'll see that they start in adults and determine if safe, they move to younger age groups. So the vast majority of the participants in these trials have been over the age of 18. So as we think about where we are today, and we think about where the trials are and where the trials have to go to truly move these new gene therapy approaches to clinical care, we must engage the sickle cell disease community to be part of the process of determining who should participate and the recruitment process for participating in these studies. So I want to share with you some data from a study that my research group conducted. We started this study in 2017, and we conducted a pilot study in April of 2017, where we conducted 3 focus groups to refine an interview focus group guide for this study, to refine a survey guide for the study, and then conducted an additional 12 focus groups. We had 3 groups of healthcare providers. We had 6 groups of adults living with sickle cell disease, and we had 6 groups of parents with a child with sickle cell disease. And we conducted this study in 2017. And I want to highlight that this is before the first gene editing trial was approved by the FDA for recruitment for that trial. And then through that process, we conducted an analysis where we identified what were the key themes for the study and issues of information and seeking information, the concerns that individuals had with regards to risk, and motivators of what would make someone participate in the trial, and the mediators that would have a factor with regards to whether they would want to participate in a trial or not. I will not go into depth with regards to this analysis now, but really want to move to really focus in on this question around information. What do people want to know about these trials to make a decision whether they have any interest whatsoever in the trial, and then ultimately to participate in the trial? So I'm going to share with you actual voices from individuals who participated in our focus groups. And by sharing their voices, I really am sharing those individual voices, but themes that we heard from a number of the participants. So these are really illustrative of what we heard across these groups. And so to frame why this issue of the need for engaged educational materials is so important. So first quote, what are the different side effects for the different types of sickle cell that you have? One patient asked with regards to the therapies. Another, an adult with sickle cell disease, asked, I want every risk associated with it. Like, even if it didn't happen, I wanted the theoretical risk. What might happen? What may happen? He said. The next person, a physician in one of our focus groups, what does it actually do for them on a daily basis? Will they have less pain? Will it halt like renal failure progression, plus any complications, the physician said? Another adult with sickle cell disease said, I'm not clear on how exactly it works. Is it something they do in a thing and then they put it in your body? How does that work? Do they inject it in your bone marrow, in your blood? How does the CRISPR get on your DNA chain to make the changes? And this was a adult living with sickle cell disease talking about gene editing and CRISPR gene editing and asking these questions. And another adult said, I want every risk associated with it. Like, even if it didn't happen, I want the theoretical risk, what might happen, what may happen. I think this is the second time I'm giving you that quote, but I actually think this is a great quote to emphasize that the individual adults living with sickle cell disease wanted to have clarity about all of the risk and the potential benefits of the treatment. As you see, this is a very well-educated group, and it's not that we don't understand or that we're resistant to research, but you need to explain in a way that people will understand. Also be a credible source. So a message here with regards to how information is communicated is extremely important with regards to the willingness to investigate these clinical trials and whether they're appropriate for their treatment and willingness to participate in the research. So I want to take a minute and talk about curative therapies for sickle cell disease. And I think this is a conversation that deserves its own webinar, just to talk about this concept of what do we mean when we talk about curative therapies? What will a cure for sickle cell disease mean? And you see here in these two examples from different media around trials that are going on and this statement about a cure. And so at 16, she's a pioneer in the fight to cure sickle cell disease is one statement. Could gene therapy cure sickle cell anemia is another statement. The question of what will a cure for sickle cell disease mean, I think is extremely important. So does that no longer mean you have a sickle genotype, or like should no longer have SS? Is that what it means? Does it no longer have any sickle cell related health complications? No longer have any clinical or psychosocial effects of sickle cell disease? So the question of what a cure means, I think, is something that must be part of the conversation and part of educational materials to help people understand that complications in organ damage that has already happened in an individual's life will not stop because of a cure. And how we describe and what we think a cure is may be different for different patients. And so exploring how do you communicate that to the community, to families, to individuals is an important part of the educational process. So how do we ensure the sickle cell disease community has access to quality, accessible, and scientifically accurate information, I think is an important research question, important policy question, and an important clinical question that must be addressed. And I would argue, and what I would like to, as we move into the second half of our presentation today, is to think about this in the context of what should the sickle cell disease community across the United States and globally should be doing to create innovative educational materials that align with the innovation that has occurred with new gene therapies. And to make sure that the educational materials and the informed consent process is not a 2000 data from a perspective of education approach versus a 2021 approach for the gene therapy treatments. That we need to make sure that the educational materials are just as innovative as the new treatments for sickle cell disease. So, what do we do when we're looking for educational materials today? We have really truly moved beyond the brochure that is sitting in a physician's office that provides information. That too can still be extremely valuable, but today, when people are seeking health information, they go to the web and they will Google and put in looking for the kind of information that they want. And so we need to understand what is on the web that's available for the community, and is it high quality or not? And so this is an issue that my research group is really interested in, is to assess the quality of the materials that are available and whether the materials are accessible to individuals and what can we do to make materials more innovative and new approaches of learning and understanding what these different gene therapy approaches are, what are the risks, and what are the benefits, and exploring these issues. I share with you here just images from various websites of information around gene therapy on the web. And you see here, it includes things from NIH, but also from other organizations where people are providing information for the public with regards to new gene therapies, thinking both about gene addition and gene editing therapies. So my research group decided to actually look at this from an educational perspective of trying to assess these materials. So we have conducted a preliminary study to assess the quality and the accessibility of these materials. So we searched the web for widely available sickle cell disease gene editing educational materials, as well as gene therapy educational materials, including videos, web pages, infographics, for the purpose of sickle cell disease populations about gene therapies. And we excluded certain things. We excluded podcasts, news articles, some of the infographics that were not educational infographics, and news articles and scientific articles. So those things were excluded from our study. And I just want to highlight here, that doesn't mean that those are not important educational materials, that podcasts can communicate information in a way that maybe other types of approaches of videos or web pages cannot. And it's important for us to think about all the types of educational materials that clearly exist. But for the purpose of this study, we wanted to limit it to those resources that were clearly developed to be educational resources. And we assessed them, using the Agency for Healthcare Research and Quality, or AHRQ, Patient Education Materials and Assessment tool, to determine with regards to the understandability and the actionability of the material. So we evaluated using the guidelines from AHRQ as part of our process. But not only the ARC PMAT understandability and actionability assessment we conducted, we also looked at the scientific accuracy. And we had scientists who are experts within gene therapy to assess the quality of the materials, the accuracy with regards to both the presentation of risk and benefits. And then we also looked at readability. And so looking at the literacy level and the ability to assess the material from a readability perspective. So I'm just going to share with you some data from some preliminary analysis that we did on using these three areas: actionability and understandability, using the PMAT, the Patient Education Material Assessment Tool, readability, using the Flesch-Kincaid grade level assessment, and the simple measure of gobbledygook or SMOG, which is also a measure with regards to readability, and then accuracy, which was based on scientific accuracy, where our reviewer looked at the materials and reviewed them for the accuracy of what was stated and what was missing from the materials. And you see here that with regards to the data of 11 resources that we reviewed, those 11 resources that we reviewed, with regards to understandability, it was generally high. It was about 72%, but actionability was fairly low. And so that raises questions from a perspective of both researchers who have clinical trials and information about where to go to get information if you're interested in a clinical trial, or how is that approached. And so it's clearly an area where I think that there's opportunity for development. With regards to readability, the readability grade levels were fairly high. The SMOG had it at 13.7, so that's grade level 13.7 or 14, so college level readability. And then the Flesch-Kincaid was at 12.0, or senior in high school readability level. And then the scientific accuracy assessment, there was clearly some variation with regards to the accuracy of the materials. So I just share this to say that there is work to be done for new materials. And I want to just stop and say there are materials that are being developed, and there are some very, very good materials that have been developed, particularly over the last 18 months. But the question is, can we do more? Can we do more with regards to innovation, with regards to these new resources and educational materials to support gene therapy clinical trials? So what we want to talk about is something that we're really interested in as a group, and that is engaging the community in the development of innovative materials. And I want to just raise a few questions for our conversation today with this webinar. How can we create high quality educational materials that can both capture the innovation behind gene therapy and engage the community in a thoughtful manner to support informed decision making? Second, how do we balance providing information about the concepts and the risk and benefits of gene therapy, while also supporting people to make an informed decision for themselves or their child? And then finally, what new strategies and approaches do we need for fully engaging the community in the development of materials? So these are some questions that we have that we hope that we can have some Q&A about with regards to creation of community engaged materials. But we put this in the perspective of what has happened over the last few years around communicating gene therapy approaches and some of the challenges that have happened. So there has been some really exciting and important work going on. And I just want to highlight a couple things. One, Dr. Lori Crosby and her group did some work with regards to looking at patient perspectives on gene transfer therapy for sickle cell disease. And one of the areas that got highlighted in their study was with regards to the use of HIV as a vector. And you see this quote from their article: participants felt that describing details about the HIV vector created more questions and uncertainty about gene transfer therapy. And so this issue of the concern of perceived risk. Another example related to that goes to the 60-minute piece that was done on gene therapy, where after the piece was presented on 60 Minutes, there was a conversation in social media about this question of how HIV as a vector was used, and are the scientists giving patients HIV. And so this is extremely why it's so important for us to create really quality educational materials and messaging on how to talk about these issues with the community to move this work forward. And there's a lot of exciting things happening within biomedical research with regards to studying innovation in educational materials. And I just want to highlight particularly the All of Us program at the National Institutes of Health, which has been quite innovative in both their informed consent process and the educational materials that they provided with regards to individuals' willingness to participate in the All of Us research program. And I think that sickle cell disease and the gene therapy industry and researchers can learn a lot from learning from innovative tools that are being created. And I give you here a couple other examples of innovative tools and innovative research going on around the creation of educational materials in different formats. And so I argue we must do that with the field of sickle cell disease. And so this question about innovation is not just the educational materials, but also the informed consent process. And can we establish an informed consent process that will help individuals in understanding the risk and benefits of a treatment in a way that is clear and that individuals clearly understand those risks and benefits? And there's a lot of work going on with this with regards to making sure that the consent process for these gene therapy protocols is extensive, is over time, and meets the needs of the individual study participant. But there may be some things that we're not doing that we could do, and things that we can study as we are studying these new treatments. So thinking about these trials that exist, can we actually conduct educational resource studies aligned with these to better understand how participants understand the risk and benefits and make their decisions to participate or not to participate? And so we can extend on the important work that's already been done with regards to informed consent and gene therapy, with regards to sickle cell disease, and informed consent and gene therapy generally, and thinking about gene therapy and its use in treatment and in clinical trials. So we argue as a research group, this should be done in a way that really is providing access to everyone. And so what we call democratizing information and building on a platform of work that's going on across this country and across the world with regards to open educational resources, that we could actually do this with gene therapy. And so providing teaching, learning, and research materials in a medium and digital or otherwise that reside in the public domain or have been released under an open license that permits no cost access, use, adaptation, or redistribution. So where the materials that are created are high quality, are scientifically accurate, but can be used by an advocacy group, can be used by a healthcare provider, can be used by a support group, can be used by a clinical trial that's being conducted. So how can we have materials that are used in this approach where they are open access and available to everyone? So democratizing information requires us to think about a number of issues. Important to make information about gene therapy publicly accessible, online, understandable, and independent of a single company or researcher's interests. And making gene therapy educational materials open source will democratize this information and create global levels of access. And so we see this as an important area of work for the field. And that sickle cell disease and sickle cell disease gene therapy clinical trials and treatments are in a great position to move forward the democratizing of this information. But there are challenges, and one challenge is how to maintain the scientific validity of the materials and how to update materials as science advances and sustain them over time. So there's a recognition that there has to be resources and support, but there also needs to be the ability for materials to be available for use in different contexts. And that takes us to thinking about this in a much more global way, with a recognition that the real burden of sickle cell disease is not in the United States, it's not in Europe, it's not in Canada, but is truly in sub-Saharan Africa and India. And as we look at the deaths with regards to children with sickle cell disease in sub-Saharan Africa, the inadequate access to the current treatments, and we're thinking about these new novel treatments, how do we make sure that just as we develop new approaches to provide access to these new treatments in sub-Saharan Africa and India, that we at the same time provide access to innovative educational materials and resources. And so a recognition that by 2050, there's expected to be an increase in the number of people living with sickle cell disease globally by more than 30%. And looking at this cartoon, you see that the major burden of sickle cell disease is in Africa and India. And so we must not just think about the European and the US experience, but also the global experience. And this ties into the work that NIH and the Gates Foundation is supporting of developing new in vivo vectors that potentially can be used in some low resource countries, and particularly in sub-Saharan Africa. And as that work moves forward, we argue educational work must also go forward at the same time. So here, the NIH and Gates Foundation is working both on HIV and sickle cell disease with regards to new treatment therapies, particularly in vivo delivery approaches. So I'm coming to a close, and I want to share another quote from our study that we conducted. And I want to just take this quote from this patient and have everyone just think about it. This patient said, "I would want them to remember that they are doing this for real live humans. These peoples have lives, families, and that this research should be conducted with care and consideration of those they are trying to benefit," a patient said. And that was a message to the research community that patient wanted to share, that a recognition that these new novel gene therapies are exciting, are important science, and is moving forward in an innovative way, new treatments. But this is ultimately for the lives and families of individuals, and that we cannot lose sight of that as we move forward on these new approaches. And that is why it's so important that we think about the educational materials. So my group, my research group, is now establishing a new engagement that we invite you to be part of to work with the sickle cell disease community in creating innovative educational materials that would be open source educational materials that can help inform and be part of the process of helping people make decisions about whether they are interested in participating in these new and exciting gene therapies. And we are interested in having you reach out to us. This is my email, bonhamv@nih.gov, to learn more about our work and what we're doing. We are now in the IRB process. So this is a process that would be a research study, but also the creation of new educational materials. So if you have interest in learning more, please reach out to us. So with that, I just want to have some acknowledgments. I want to thank all the study participants who shared their voices that were part of our research study. And I want to thank John Tisdale, Dr. John Tisdale, Dr. Alexis Leonard, and Kiana Amini, and Courtney Stewart, and the Bonham Lab. Thank you.
Wonderful. Thank you so much, Mr. Bonham and Ms. Amini, for your presentation. That was really insightful as well as powerful with those patient messages. So at this time, I want to remind folks that we're coming to the question and answer portion of the webinar. And so we'll begin taking questions through either the chat box or question function of the control panel of this webinar. So if you have any questions, feel free to put them in those spaces now, and we'll start looking for them. Susan, did you have any thoughts, comments, or see any questions coming through?
I'm going to. So many thoughts and questions. I'm just this exceeded even my very high expectations, and really, really grateful for your time putting this presentation together and sharing it with us. The first half of my career was in innovative educational technology and presentation and how to get complicated information to a meaningful level. And so I'm super excited about this and a lot of the things that you were talking about. I did want to, you talked a little bit about what you're starting to do with your group at the end, and I wanted to see if we could dive a little bit more into that and talk about the kinds of things that you're working to develop. And in particular, one of the things that interested me, let me back up. I have done the consent process for the All of Us research study, and have to say it was just mind-blowing as someone who's worked on issues of consent and patient education for a long time. I just thought it was beautiful. And there's so much of that we can, I think, draw into the work that we're doing and into other areas. So I wanted to ask if you could frame a little bit what kinds of products you're working towards, what kinds of modalities of education that you're working towards. And also, one of the things that I was curious about was thinking about ways to incorporate, and I think all of us did this to a certain extent, ways to incorporate people's understanding of the material that they're getting before they get to the point of trying to make a decision around consent. So that's a lot, and you can pick bits of that to talk about.
Yeah, I'll take some bits of that, and I'm going to ask my colleague also if she has any comments after I talk. So what we are doing is organizing a process to bring different stakeholders within the sickle cell disease community together to develop new materials and to agree on what should be included. And that's including the various constituencies within the sickle cell disease community, but where the core of that is the patient and the family. And so using that process of having industry and government and healthcare providers and the researchers who have developed within academic institutions these different trials and treatments, engaged with advocacy organizations and with patients in actually organizing, developing, and designing materials. And so that's the first step of our process. And then taking that step on how do you create them in an innovative way with regards to approaches. So that's the work that we're doing, and really interested in this process where it's not just one company or a government agency with 2 or 3 individuals who may be living with the disease, or family members that create the materials, or an external group that creates the materials, but actually a deliberative process of engagement with regards to creation of content that then can be open source and available to everyone. So that's the framing of the work that we're doing. Kiana, is there anything you would like to add to that?
Yeah, I'll just add that I think in addition to using this process to figure out and explore what different kinds of mediums we want to use to create different educational materials, I think another hope of it is that we can also use this process to understand what is the best way to communicate and message and frame these kinds of concepts. And this goes back to Mr. Bonham's slide about the challenges with communicating gene therapy approaches, with concerns about, with the use of the HIV vector, and there's other kinds of things that I've heard a lot of patients want to know more about, and also going back to the whole question of how do you frame this as a cure when we don't even know what a cure means. And so I think what we hope to gain from this process is just an understanding of what is the best way to present this information, what is the best way to frame every part of it, every risk that comes with it, every benefit that can come with it, in a way that's thoughtful and will support people to make the best decisions for themselves. So that's what we're hoping to gain from this process.
Thank you. And so the process itself is that you're essentially asking for input from as many different community-based, advocacy-based groups and individuals as you can, and sort of just drawing folks into conversation. Will you be having meetings, webinars? Can you talk a little bit about what it'll look like?
Yes. No, we're seeking to bring, within various ways, and now, of course, with the pandemic, that many things are happening in this virtual world that we live in, but to bring the different constituencies and stakeholders together in a process over time to develop materials. So it is what we describe as a deliberative process that is not like a one-time focus group, but our ability to work with a group of individuals that are committed to be part of this process over a period of time to develop both content, and then working with various experts around new innovative approaches.
Thanks. And then that information that you've gathered, is that, do these materials then, like I'm thinking of what you mentioned at the beginning, what you said at the beginning is really who should be the communicator, how do people receive messages from different entities. And so would these materials that you're developing and approaches that you're developing, then like how would the voice come from them to the people receiving them, to the people doing the learning?
Yeah, so we want to work with the community with regards to various venues and ways for different constituencies within the community to have access to them. And this gets to this issue of open source with regards to resources and how they're able to be used in various ways. And so, I think one of the exciting things coming from the National Human Genome Research Institute in the field of genomics, where that there's a real push with regards to the whole community that to make sure that data and materials are available and open sourced in ways. And we see similar kinds of approaches that can be used around education to push forward innovation with regards to educational materials. And so that is a part of the model.
I'll also add that I think as I've become more immersed into this work, I've been learning about how the wide sickle cell disease community is not very well organized and coordinated in terms of like different stakeholders kind of being very separated and in different kind of corners doing similar work that could be even more strengthened if people could come together. So I think the hope is that with this process of us gathering different people from different areas of work and expertise, is that this is also an opportunity to create some kind of network that can then work together to bring the community together and integrate different agendas and interests in a way that will be hopefully sustainable through our educational materials that we'll create.
And we just want to highlight, we recognize that there is important work going on and people are coming together, and that, you know, that is happening. But I think that there's also opportunities to kind of take it to the next level with regards to some of this engagement in a way of creating new innovative and new types of approaches and new ways that have been thought about of ways of sharing materials, as well as thinking about those programs like the All of Us program that have really helped us set where we are with regards to innovation to make sure that that's happening within the sickle cell disease community.
Thank you. We have a couple of comments and a couple of questions coming in. One is, and I think I know your answer, but you might want to highlight it a little bit. The question is, do you feel having patient engagement teams is necessary to help with educational documents and clinical trials? It sounds like that's where you're going.
Yeah, the answer is definitely yes.
Yeah.
I think, you know, without having families and patients, that there will be a failure to understand what the issues are from their perspective, and that their involvement and partnership with them is essential for the creation of innovative materials. You know, the researcher or the company may know the science, but may not understand the experience of that individual or that family, and that's extremely important with regards to the development of materials.
Thank you. A question from Tessa at American Society of Hematology, who wonders if there is a timeline, and you talked a little bit about the structure, so a timeline and structure for the development of this work. She said that the larger sickle cell disease coalition holds regular meetings with stakeholders and could offer these meetings as a forum to engage insight.
No, I think that there's really some real great opportunities for collaboration. You know, I've had some conversations with some of the folks at ASH about this work. So, you know, look forward to opportunities. We see this project moving forward this year.
Okay, thanks. And a comment, thanks for this great talk, Dr. Bonham, very interesting work. I think it will help get wider participation from other sickle cell disease populations as gene clinicians and CDO leaders are involved nationwide, and that certainly seems the way that you're going, and it's very exciting. And then we just got one more comment in. A suggestion to reach out to the 5 HRSA Sickle Cell Disease Treatment Demonstration Project regions, and a number of them actually are on this call, and Kim Morrison at HRSA about this work, and again, another set of potential collaborations that I think could be generated out of that.
Yeah, and I would just want to again give my email address. It's Bonham, BONHAMV, at nih.gov. You know, please reach out to us also if you're of your interests and engagement, and as well as we clearly will be reaching out to various constituencies that have been identified.
Thank you. And I'm going to give it just maybe another 30 seconds or so to see if any other questions come in. But in the meantime, I just really want to profoundly thank you. And in addition to following up with your email, is there a website or anything like that that folks can go to sort of keep track of the progress of this work?
That's a great question. So right now, no, but that may be something that we can actually establish, some web pages that would be on our website at genome.gov about the process as it moves forward. But today, there's not a website.
Okay, that'd be, if you do come up with something, let us know, and we will let everyone who's here and everyone on our email list know about it. Just a couple more comments saying how helpful this presentation was. So I'll sort of lump them together, and just great appreciation to you both for this presentation and for this work.
Great. Well, thank you, and thank you for the invitation. And please, again, we would love to talk to everyone who's interested in this topic, as well as to engage with those that are doing work, because I know that there is work going on. So thank you very much.
Thank you. And Niani, do you have finishing remarks?
Sure, just to go ahead and close it out and echoing everyone's sentiments about how interesting and wonderful this presentation was, and looking forward to seeing the future of this work. So we'll conclude by just sharing that to reach out to us. I think most folks are on our webinar list, but if not, we can be found for these webinars and continuous information like this at trackingcalifornia.org, as well as Sickle Cell Resources on Facebook, as well as casicklecell.org. And we post our webinar recordings to both our Tracking California website as well as casicklecell.org. So this will conclude the time that we have for this session. Thank you so much to everyone again who participated. And thanks again for joining us today, and we'll see you next time.
Niani, I'm going to stop recording. Is that okay?
Yes, that's fine.
Okay, great.
Recording?
We have stopped recording.
Okay, well, good. Was that what you wanted? Or?
That was just fantastic. Thank you both so much. You touched on so many things that sort of come up for me as I'm listening, and it's, you know, it's not about me, but as